Om Swastiastu. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya, and best wishes and regards to all of us. Welcome to our presentation. In today's occasion, we will be presenting about our student project called Central Dogma in Molecular Biology, presented by S.Ked B1. Next slide. Here's the list of our members from S.Ked B1. You can pause the video to take a look. Next slide.
Thank you for the time given to me. Now I'm going to explain about the background of our student project. But before that, let me introduce myself. I'm Sienny and the last three digits of my student number is 004. In this presentation, we're going to talk about a theory in the study of molecular biology named the Central Dogma. This term was first introduced by Francis Crick in 1958 to describe the flow of genes from DNA to RNA and RNA to proteins. Over time, scientists have found a few contradictions towards the Central Dogma, which are retroviruses and prion disease. Therefore, it is important to learn about the Central Dogma, DNA, RNA, proteins, and mutation to better understand their influence in molecular biology. That's all from me. Thank you. Next slide.
First of all, let me introduce myself. My name is Niputusinti Haramrahyatak with 3 last digits. Student ID is 018. Now, I will mention about problem formulation, objective, and benefit of the student project. The problem formulation are what is Central Dogma and how is its history, what are DNA, RNA, and protein, how is the process of DNA replication and protein synthesis, what is genetic code and what is its role, what are the exceptions to Central Dogma, and what is genetic mutation and how does it affect the genes in DNA. Then, the objective of the student project is to describe the Central Dogma as an important theory in the process of changing genes from DNA to RNA and RNA to protein by explaining definition, history, process, and structure that involved in Central Dogma theory. And the benefit of the student project, the author is expected to add insight into the Central Dogma, both the structure involved in it and the process associated with the Central Dogma. For other writers, it is hoped that it can be a reference for writing future study. Next slide.
Hello, my name is Lichana Bianca Kusuma with 3 last digits of student ID is 025. Definition of Central Dogma. The Central Dogma of molecular biology is a theory stating the relation between the main informational macromolecules such as DNA, RNA, and proteins. The canonical interpretation of the Central Dogma is that genetic information, DNA, is utilized to generate transition messenger molecules that are used to direct the information needed to make proteins to the ribosome. The ribosome serve as factories in the cell, where the information is translated from a code into a functional product. The process by which the DNA instructions are converted into a functional product is called gene expression. Gene expression requires 2 processes, i.e. transcription and translation. That's all from me. Next slide.
Hello, my name is Ananda with the 3 last digit of student ID number is 021. So today I'm going to present about history of Dogma Central. The phrase comes from a seminal paper on protein synthesis presented by Francis Crick at the 1957 annual meeting of the Society of Experimental Biology and published in 1958. In this work, Crick lists a standard set of 20 amino acid residues, of which for the first time, the specificity of the fragment of nucleic acid is expressed only by its sequence of bases, which is in the amino acid sequence of a given protein. The code posits that the 3-dimensional conformation of protein must be determined by its amino acid sequence, points out that protein synthesis must occur sequentially, and presents the hypothesis of an adapter. a molecule that mediates protein formation on a ribosome. Most importantly, it formulates what Crick called the Central Dogma. There's no manuscript of Crick's actual talk, but according to recognized in the paper, the version that we are all familiar, which was the product of my discussion with Sydney Brenner, who's redrafting the manuscript for the publication. Modern readers probably got disturbed by Crick's opening statement. I shall agree that the main function of genetic material is to control the synthesis of protein. There is a little direct evidence to support this, but to my mind, the psychological drive behind this hypothesis is at the moment independent of such evidence. This highlights how uncertain scientists were at the time about the gain function, because not everyone accepted that nucleic acids were involved in protein synthesis. Next slide.
Thank you for the opportunity. My name is Krisnaditya Dwi Raharja with 3 last digit of student ID numbers 014. Okay, so we are going to continue our presentation. The first one is DNA. What is DNA? DNA, or deoxyribonucleic acid, is a molecule in our body which is our blueprint for any instructions such as grow, survive, develop, and reproduce. DNA has a regular structure. It means orientation, length, and number of nucleotides is constant. DNA consists of deoxyribose, phosphate group, and nitrogenous bases. Watson and Crick were the first one to propose the double helical structure from analysis using X-ray diffraction patterns, model building, and chemical analysis. Adenine makes 2 hydrogen bonds with thymine, and guanine makes 3 hydrogen bonds with cytosine. So the amount of adenine is equal to thymine, and the amount of guanine is equal to cytosine. Next slide. Main difference between DNA and RNA is located on the helical structure. Most RNA is single-stranded. RNA consists of ribose, phosphate group, and nitrogenous bases, where thymine in DNA replaced by uracil. The amount of purines is not equal to the amount of pyrimidines. Next slide. Protein can be classified as macromolecules. Protein is polymer that consists of amino acids as the monomers. Protein has 4 levels of structure: primary, secondary, tertiary, and quaternary. That's all from me. Thank you. Next slide.
Thank you so much for the opportunity. First of all, let me introduce myself. My name is Rahel Manurung, with the last digit of my student ID 030. So now I'm going to explain in general about the DNA replications. So DNA replication is a process of copying DNA genomes and cells, and occurs during a phase of cell cycle. And in eukaryotic organism, there are 3 main stages, which are initiations, elongations, and terminations. At first, initiations. So the process of DNA replication starts with DNA that unwinds at the origin of replications due to enzyme helicase. Along the DNA opens, replication forks are formed, and there are 2 replication forks that are formed at the origin of replications, and as the replication process continues, this grows in both directions. And the second stage, which is elongations, the parental strands that complement each other receive the new bases. The new strand is created continuously, whereas the other strand is created in pieces. Last stages, terminations. The primer is removed, followed by replacing the primers with fresh DNA nucleotides, and the DNA ligase enzyme fills the spaces between the fragments. That's it for me. Thank you so much for the opportunity. Next slide, please.
My name is Arin Arnaz with 3 last digit number of student ID is 002. Although the information in RNA is copied into another chemical form, it is still written in the same language as DNA, the language of a nucleotide sequence. As a result, the name transcription. Transcription occurs in 3-step process: initiation, elongation, and termination. The RNA polymerase first recognizes the gene to be transcribed and binds to a specific sequence on DNA known as the promoter. After binding to the region of the gene that will be transcribed, the RNA polymerase continues to separate the double helix and continues down the template, making a new complementary single-stranded RNA. The single-stranded RNA is kept at 5' in which introns are deleted from the RNA, leaving only the required nucleotides for translation, known as exons. The newly produced RNA strands get to the cytoplasm via the nuclear pores. Thank you. Next slide, please.
Okay, in this section we'll talk about translation, but first let me introduce myself. My name is Ananda Aulia, with 3 last digits of my student ID is 015. Translation is the process of converting information that's stored in mRNA into proteins. Translation takes place in specialized organelles called ribosome. This process is similar in prokaryotes and eukaryotes. The first step of the translation process is initiation. Initiation occurs when the small ribosomal subunit binds with initiation factor and an initiator tRNA at the start codon of an mRNA, followed by the binding to the initiation complex of the large ribosomal subunit. The next step is elongation. During the elongation stage, a charged tRNA binds to mRNA in the A site of the ribosome. A peptide bond is catalyzed between the two adjacent amino acids, breaking the bond between the first amino acids and its tRNA. The ribosome moves one codon along the mRNA, and the first tRNA is moved from the P site of the ribosome to the E sites and leaves the ribosomal complex. The last step of translation of termination occurs when the ribosome encounters a stop codon, which doesn't code for a tRNA. Release factors cause the polypeptide to be released and the ribosomal complex dissociates. Next.
Before we begin, I would like to introduce myself. My name is Refferty, with the last 3-digit number of my student ID 003. I would like to talk about genetic code. So genetic code is a sequence of nucleotides in deoxyribonucleic acid and ribonucleic acid that determines the amino acid sequence of proteins. We generally refer the genetic code from messenger RNAs as codons. Basically, nucleotide bases of messenger RNA are adenine, uracil, guanine, and cytosine. Those nucleotide bases can be converted to triplets that will make up a chain of amino acid to form a protein. In the right screen, we have a genetic code of a messenger RNA table, which contains 64 codons. Out of all sequence, 61 of them specify the amino acids, and 3 of them are stop signals. The stop signals are uracil adenine adenine, uracil adenine guanine, and uracil guanine adenine, whilst the start signal is adenine uracil guanine or methionine. That's all for me. Next slide.
Okay, in this section we will talk about the exceptional condition of central dogma theory. Before that, let me introduce myself. My name is IG Mahaprajadipasta with 3 last ID number 028. Now let's start it. This section means that there are some conditions that actually doesn't follow the central dogma theory. What kind of condition? The first one is reverse transcription, and the second is prion disease. Reverse transcription is a process where the single strands of RNA can be converted back to bDNA, especially complementary DNA or cDNA. This mechanism needs help from the reverse transcriptase enzyme. This technique is very useful for sequencing, characterizing, and often used in RT-PCR, but many viruses also do this mechanism, for example, in retrovirus and HIV. The process of converting the RNA back to bDNA in this condition doesn't converge with the central dogma theory. Prion disease. This disease is one of the fatal neurodegenerative disease. PRPSG is known as an agent of the prion disease, where it is a disorder of the nucleic acid moiety resulting from misfolding of prion proteins. This disease happens when the autocatalytically misfolded process happens in the PRPC host that generate PRPSG infection. The PRPC host that has ability to be self-replicating, it is not in accordance with central dogma theory, because in central dogma theory, the protein shall not be replicated individually. That's all from me. Thank you.
Okay, thank you so much for the opportunity. My name is Widhanti Prabhawaputra with 3 last digits of my student ID number 022. Now I would like to explain about genetic mutations. Genetic mutations is an alteration of genetic information in an organism's cells that can be caused by either hereditary or environmental factors. Genetic mutations range from large-scale changes in DNA line segment due to chromosomal rearrangement to chemical changes in gene base pair, which we call point mutations. Cases of change in the structure of chromosome are generally caused by error during meiosis or exposure to damaging agents such as radiation that cause chromosome breakage. There are 4 kinds of changes in structure of chromosome, which includes deletions, duplications, inversions, and translocations. Next, we have point mutation. So point mutation is mutations that caused by changes in one of the nucleotide base pair in gene in DNA. Point mutations can be divided into 2 general classifications: base pair substitutions and base pair insertions or deletions. Base pair substitutions is a type of point mutations that occur due to the replacement of one nucleotide and its partner with another type of nucleotide. Depending on the location of the base pair substitutions, this type of mutations can lead to silent mutations, missense mutation, or nonsense mutations. In addition to undergoing substitutions, nucleotide pairs can also be inserted by another nucleotides, which we call insertions, or it can also undergo deletions. This category of mutations result in a shift in the reading frame of all codon strand from the insertion or deletion sites, which we call frame shift mutations. So that's all for me. Thank you so much. Next slide.
First of all, let me introduce myself. My name is Devalenzara Ramadan with 3 last number 016. So, the conclusions of this student project is the central dogma of molecular biology is a theory stating that genetic information flows only in one direction, from DNA to RNA to protein, or RNA directly to protein. Francis Crick at the 1957 annual meeting of the Society for Experimental Biology first described the central dogma as the detailed residue-by-residue transfer of sequential information. It states that sex information cannot be transferred from protein to either protein or from protein to nucleic acid. Next slide. In reality, there are several processes that are not in line with the dogma central theory, which can be divided into 2 categories. There are reverse transcription and prion disease. Reverse transcription doesn't follow the dogma central rule because it reverts back to the process where RNA can be transferred into DNA. On the other hand, prion disease doesn't follow the dogma central rule because the replication of protein is not supposed to happen in nature. There is only the replication of nucleic acid, not the protein itself. In the future, there might be a new theory that can describe the phenomenon clearly.
This is it. We have reached the end of our presentation. We hope that the audience could gain many new knowledge from our presentation. We are sorry if we have said something wrong or inappropriate. Thank you for listening, and I will close this presentation with Parama Santi. Om Santi 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 Om. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya, and best wishes and regards to all of us. See you.